Live. Hello, good evening, everybody. My name is Zaina Khalil. Thank you all for joining. I am a nurse practitioner at the UCSF Minimally Invasive Gynecological Surgery Department. Thank you for anyone also that's watched any of our webinars from last week and last night, and welcome to anyone who's joining us for the first time tonight. Let's see here. So it's my pleasure to welcome you all to this final session of our ENACT Core B webinar series. This is part of the UCSF-Stanford Endometriosis Center for Discovery, Innovation, Training, and Community Engagement, and is focused on understanding endometriosis mechanisms, improving disease classification, discovering new treatments, enhancing patient outcomes, training future researchers, and engaging the community to support research and empower those impacted by this condition. On tonight's end, we have, I know we definitely have Dr. Lager on, who's one of our wonderful program planners. She's the section chief for minimally invasive gynecological surgery and associate director of the Endometriosis Center. And I believe at some point tonight, we might have the pleasure of having Dr. Jessica Pokuanane also on the call, who is the gynecology division chief at Rutgers University and also a program planner. Specifically, in tonight's review, we'll be taking a deeper dive into the research being done on endometriosis, as well as the future of machine learning and AI to advance our knowledge and treatment options, as well as what's on the horizon in future innovations. Some housekeeping real quick before we get started. We would love to see your bright faces, so feel free to turn your video on if you'd like. However, we do please ask that you keep yourselves muted. We certainly will be welcoming your questions and we'll be taking them at the end of the presentations, but please put them in the chat or the Q&A box, and we will do our best to compile them and get to them at the end of the session. I'll go ahead and start introducing our speakers. We have Dr. Marina Sirota, Dr. Tomiko Oksatsky, and Dr. Khan. Dr. Sirota is currently a professor and the acting director at the Bakar Computational Health Sciences Institute at UCSF. Prior to this, she has worked as a senior research scientist at Pfizer, where she focused on developing precision medicine strategies in drug discovery. She completed her PhD in biomedical informatics at Stanford University. Dr. Sirota's research experience is translational bioinformatics and spans nearly 20 years, during which she has co-authored over 170 scientific publications. Her research interests lie in developing computational integrative methods and applying these approaches in the context of disease diagnostic therapeutics, with a special focus on women's health. She leads the UCSF March of Dimes Prematurity Research Center, as well as Codex's ENACT Center. Her passion for women's health and passion for research is ever more apparent, as she's also the founding director of AI for All, which is a program at UCSF introducing high school girls to the applications of AI and machine learning in biomedicine. Absolutely incredible. Similarly, we have Dr. Tomiko Ostoksky. She is associate professor with the Division of Clinical Informatics and Digital Transformation and a physician scientist in the Sirota Lab at the Computational Health Sciences Institute here at UCSF. She earned her BS in biochemistry from UCLA and her MD from the Medical College of Wisconsin. Her research involves integrating diverse data sets, including electronic health records, to better understand human health and disease. She co-leads the UCSF March of Dimes Preterm Birth Research Center and is the director of the UCSF AI for All program, teaching AI to high school students as well. And last but not least, we also have Umair Khan. He is a PhD student at the Biological and Medical Informatics program at UCSF. He studied computer science and mathematics at Portland State University and is interested in developing and applying novel computational methods to advance all our knowledge in health and disease. His work on endometriosis spans a range of data modalities, including single-cell sequencing, methylation, and as he will discuss today, electronic health records. Without further delay, I'll hand it over to y'all.
Awesome. Thank you so much for the kind introduction. I will kick us off, and then I'll turn it over to Tomiko, who will turn it over to Umair, and then I will wrap up. Let me share my screen, and hopefully this works. Okay. Let's see. Can everybody see my screen? Do I need to swap, or is it good?
Swap it. Yeah, it's
Swap it.
on the speaker
Okay.
view.
Thank you.
Sure.
Always a challenge. All right. So we're going to tell you today about how we leverage data, computation, AI in the context of endometriosis, and share some of the work that we've been doing as part of the ENACT Center at UCSF. So I'll start by saying, probably many of you are familiar, but endometriosis is an inflammatory condition that's characterized by the growth of endometrial tissue outside its normal location, so where it's not supposed to be. Some common symptoms of endometriosis include pelvic scarring, pain, and infertility. It's very difficult to diagnose and treat due to the fact that this disease is so complex and heterogeneous. And current treatment include pain medications such as ibuprofen or naproxen. Oral contraceptives are often used, which have their own side effects, desired or not. And of course, laparoscopic surgery in more extreme cases, which is very invasive. So in our group, we figure out how to use data, whether it's molecular data, which could be gene expression or genetics or antibody repertoire, epigenetics, microbiome, the metabolome, or proteome. And many of these measures now we can have on a single cell level. And also we use electronic medical records data, which is information that gets captured when somebody goes to see a clinician, whether that's diagnosis codes, lab test results, encounters. And we have an incredible resource at UCSF and across the whole UC system. And Umair, well, I guess both Tomiko and Umair will touch upon how we leverage this resource, which is de-identified clinical data on all of our patients that we can leverage for research purposes to ask various questions. And bringing the molecular data, either from the public domain or data that we generate ourselves with the clinical data, together with advanced computational approaches, as well as AI, we have a unique opportunity to ask new questions about disease and look at disease from a different lens than we've been able to in the past. And in terms of computational approaches, I just wanted to set the stage and say that there's different ways that we can analyze data. There's traditional statistics that allows us to differentiate what signals might be significant or not, what's different between cases or controls. There is traditional machine learning approaches which allow us to predict what might happen to a patient down the line. And then now there's these generative AI approaches, which we're not going to talk about today, but there's a lot of opportunities to leverage them, which allow computers to process text and interact with individuals in a much more human way than machines could in the past. So putting all this together, we have an incredible opportunity to try and leverage computation and AI across scales. And this slide is borrowed or was created jointly with Dr. Tony Capra, who is a faculty member at UCSF and also one of directors at the Baker Institute, specifically for AI strategy and education. And we've been thinking a lot about how we can look across scales, starting from DNA to protein to cells to tissues to physiological models to finally models that allow us to look at patients and apply computation and AI across the scales. Because of the heterogeneity of endometriosis as a condition, and I believe you might have heard from Dr. Jadis yesterday or the time before, we collaborate very closely together. This is a paper led by Dr. Skotsky, but in collaboration with many of us that have explored all of these multi-omics and different type of data approaches in the context of women's health. And we really think they can be instrumental in having an impact for endometriosis because it's such a multifactorial condition. So with that in mind, the ENACT Center was launched several years ago. It's co-led by Dr. Tadis, myself, and Dr. Garnier at Stanford. And if you're interested in checking out the website, this is it. And we have 3 different projects. One focused on leveraging single cell signals and understanding endometriosis from a single cell perspective, which is Project 1, led by Dr. Jadis. Project 2 explores environmental factors and ways that they might play in the context of endometriosis. That's led by Joshua Robinson, Dr. Joshua Robinson, Dr. Susan Fisher, and Dr. Tracy Woodruff. And then Project 3 is around computational drug repurposing. And we will start by talking, which is led by myself in collaboration with Tomiko and as well as others. We'll start by telling you a little bit about the type of work that we do. This whole center is an incredible effort, has a number of collaborators, and really leverages some of the biobank resources that Dr. Jadis has put together over the years. We also have an educational and an administrative core, and we collaborate with many groups across UCSF, Stanford, Rutgers, and beyond. So zooming into Project 3, these are our overall aims. We leverage gene expression data to come up with therapeutic predictions. We aim to validate those in human cells, and that work is ongoing, and then also explore them in animal models. And I will turn it over to Dr. Oskotsky to tell you more about what we do.
Great, thank you, Marina. So as we know, taking a drug, developing it from scratch, and looking to see if this is something that's effective to treat a particular condition takes many, many years, roughly up to 15 years, as well as a lot of financial resources, up to $800 million just to bring a novel drug to market. Sadly, 90% of drugs fail in early development. So there has to be other ways that we can try to identify therapeutics in order to treat conditions. So next slide, please. Yes, so one of the solutions is drug repurposing. This is a way of identifying drugs that are currently FDA approved that might be able to be used for conditions other than those that they were originally designed to treat. By doing this, we can not take as much time to identify drugs and also reduce the risk of failure. Next slide, please. So computational drug repurposing is one way of identifying drugs that can be used for other conditions besides those they were designed to treat. And there are many ways that this can be done, including target-based drug repositioning, disease-based, molecular modeling approaches, network approaches, as well as global signature-based approaches. For complex diseases, the one drug, one target, one disease paradigm may not be sufficient, in which case novel single or combination therapies identified by drug repurposing methods may be promising. Next slide, please. Next, I'll talk a little bit about sources of data that can be used for computational drug repositioning. One is on the disease side, there are repositories like GEO, which stands for the Gene Expression Omnibus, that have publicly available gene expression data for many conditions and diseases. On the drug side, there are collections like the Connectivity Map, or CMAP, as well as links that have gene expression data from cultured human cells exposed to FDA-approved drugs, as well as non-drug bioactive small molecules. Next slide, please. For our work, we leverage a computational drug repositioning pipeline that was pioneered by our co-presenter today and one of the PIs of our ENACT Center, Dr. Marina Sirota. And this here reflects a signature-based approach for drug repurposing. We use the gene expression data from those with a disease of interest and those without. And from this, we identify differentially expressed up and down regulated genes to use as our input disease signature, as reflected on the top left of this slide. We also use the gene expression signatures of drugs, as also reflected on the top left. And for each drug, the differential expression of the shared genes is compared to our input signature. From this, we calculate a reversal score using a non-parametric rank-based method based on the Kolmogorov-Smirnov test. Our hypothesis is that if compounds reverse the disease signature, then they may be therapeutic, potentially, for the condition. Next slide, please. And here we see that this method has been successfully applied in previous work exploring therapeutics for conditions including inflammatory bowel disease, preterm birth, and COVID. Next slide, please. Here we see an overview of this approach applied to endometriosis. Specifically, we used bulk gene expression data of eutopic endometrial samples that were collected from women with endometriosis and those without disease to determine our input signature. And this data was originated from the lab of Dr. Linda Dudies, one of the PIs of our ENACT Center, and is shared publicly. We first normalized this data and batch corrected it. Then we looked at differentially expressed genes of disease versus control of all the samples together in an unstratified fashion, and also stratified by disease stage, that is ASRM stages 1 and 2, or stages 3 and 4. We also stratified by menstrual cycle phase, proliferative, early secretory, or mid-secretory. Differentially expressed genes that passed our cutoff of an adjusted p-value of less than 0.05 and log fold change of greater than 1.1 were considered significant genes for each signature. On the drug side, we used CMAP to obtain gene expression profiles from the cells that were treated with with drugs. And for the unstratified and 5 different stratified endometriosis signatures, we calculated reversal scores as described earlier for each drug based on the data from CMAP. Next slide, please. So on the top right of this slide, we see an upset plot that shows the number of drugs that were identified by our 6 endometriosis signatures. There were 299 unique drugs identified by at least one of the endometriosis signatures. And we see reflected by that bar on the far left of this upset plot, 221 of the 299 drug candidates were commonly identified across the 6 endometriosis signatures, which showed consistency in the majority of the drug candidates from the unstratified and stratified signatures. The heat map on the left shows that the endometriosis disease, endometriosis gene expression signature, say that 5 times fast, on the far left, and the gene expression signatures for the drug candidates with phenoprofen, the drug having the greatest reversal score, that's on the left. And then as the reversal scores go down, those drugs are on the right. On the bottom, what we see is a heat map of the reversal scores from our computational drug repositioning pipeline for the 6 endometriosis signatures. And the top 20 drugs from the unstratified signature in order of reversal. The darker the purple, the greater the reversal. And as you can see, phenoprofen, the top candidate, which is at the top, shows the greatest reversal across all 6 disease signatures. Next slide, please. So here, what we see is a network diagram that just shows that first of all, this use spoke is a knowledge network that includes information from drug bank and Chembl databases. We looked at the top 20 drugs from the unstratified signature. And what we saw is that conditions associated with inflammation in the immune system, cardiovascular system, neoplasms, infectious disease, and psychiatric conditions were all those that these drugs affect. And again, we see that some of these have one target, and others, like promazine, may have a couple, or some of these conditions may have more than one drug that target them. Next slide, please. So going back to phenopropin specifically, this drug is the top therapeutic candidate that was identified from our unstratified signature, and it was among the top, if not the top candidate from the stratified endometriosis signatures. We wanted to look into this drug particularly because it just seemed so effective. NSAIDs, phenopropin is an NSAID, which is a class of anti-inflammatory drugs that's commonly used for treating endometriosis and other conditions associated with pain. It's a drug that's been available since the 1970s, although it still requires a prescription to this day in order to use it. From a clinical trial back in 1985, phenopropin was shown to be better than aspirin at treating primary dysmenorrhea, although the study had a small sample size with only about 90 or so patients. So really to date, there have been no studies looking at phenopropin as a therapeutic for endometriosis. Next slide, please. So here, what we did was we looked at the electronic medical records across our University of California healthcare institutions, just to see what the use of phenopropin was like, and as well as NSAIDs in general. This data includes records from UCSF, as well as UC Davis, UC Irvine, UCLA, and UC San Diego, with data going back as far as 2012. And interestingly, of the over 60,000 patients who were diagnosed with endometriosis, chronic pelvic pain, or dysmenorrhea, we see that well over half of these patients were prescribed any NSAID. However, just 5 or less than 0.01% were prescribed phenopropin. Next slide, please. So to validate phenopropin as a potential therapeutic candidate for endometriosis, we used an animal model of endometriosis, which was work led by Stacy McAllister and her team at Emory. We also used, in addition to phenopropin, we used ibuprofen as a positive control, as well as no surgery and no treatment as negative controls. In this work, the rats underwent surgery to implant pieces of their uterus in their pelvic cavity. And then vaginal hyperplasia, or an increase in vaginal nociception, develops and stabilizes in about 10 weeks. Volumes of water are then delivered to the rat's vaginal canal, and the rats are trained to poke their heads into a tube in order to stop the vaginal distension. By poking their heads, they break this beam of light, and then the water that's delivered to the balloon stops. Next slide, please. So what these graphs show is the rat's escape response on the y-axis at the different volumes of water. And we see on the x-axis is so the escape response is on the y-axis, the water volumes are on the x-axis. The blue line that we see in the graph represents the pre-surgery or baseline period responses. The orange line represents the escape responses after the endometriosis surgery is done. And then the green line reflects the post-treatment period. And as we see here, the escape responses post-endosurgery are greater at each volume of water tested. However, treatment with either phenopropin or ibuprofen bring the escape responses back to what they were during the baseline pre-surgery period. These tables reflect p-values and Bonferroni adjusted p-values from Mann-Whitney U-tests. Basically, there's a significant difference in the escape responses between baseline and post-endometriosis, as well as post-endometriosis surgery and post-treatment. but there's no significant difference in the baseline and post-treatment periods for both phenoprofen and ibuprofen. Next slide, please. So to summarize, we used a computational drug repurposing method to identify treatments for endometriosis. From our work, the NSAID phenoprofen emerged as having the greatest disease signature reversal among the drug candidates. And our analysis of the University of California-wide EHR data showed that while NSAIDs are commonly prescribed to endometriosis patients, phenoprofen is not. In our animal model validation work, we found that phenoprofen alleviated vaginal hyperplasia in a rat model of endometriosis. And in future studies, we could explore whether drug candidates for endometriosis from this work, or other drug candidates from this work, as well as potentially use single-cell transcriptomics data to identify novel single and combination therapeutic strategies for endometriosis. And now we will hear from our very talented graduate student, Umair Khan, about his work with electronic medical records.
Cool, yeah. Thanks for the introduction, and yeah, thanks for the opportunity to share some of the work we've been doing here. So yeah, as Marina and Tomiko have talked about, we here at UCSF have access to a really incredible amount of data through the electronic medical record system. So we have access to complete records from at least 2012 onwards. And in fact, we have converted records that go as far back as the 1980s. So in total, we're talking about tens of millions of records. And this includes things like diagnoses, encounters, lab results, medications, and procedures. And all of this is incredibly useful when it comes to understanding complex multifactorial diseases like endometriosis. Next slide. So yeah, as we've talked about, there's an increasing understanding of endometriosis as a multi-system disorder with very wide-ranging effects. So here I've just highlighted a few of these studies over the past decade that look at links between endometriosis and things like various autoimmune diseases like MS or lupus, ovarian cancer, and even chronic diseases more generally. So the question sort of then becomes, how can we, first of all, just describe the clinical effects of this very complicated disease that seems to be associated with conditions all across the body? And then secondly, can we use that information in some way to better understand how to manage it or treat it? And yeah, next slide. So, as you might have guessed from what we've talked about, electronic health records provide a great starting point for characterizing and understanding all this heterogeneity. So here I've highlighted a couple of previous works that use electronic health records or EHRs to look at the comorbidities of endometriosis, either broadly, but in a relatively small patient population, as with the first paper on the left, or in a large patient population, but at a very specific class of mental health conditions in the second paper on the right. Since we have access to EHR data at UCSF, we can explore this question at a previously unseen scale. And we go about doing this in a couple of ways. So first, we use the diagnosis records to find patients who have endometriosis. And then we kind of branch off into two separate analyses. So in the first analysis, we compare our endometriosis patients to control patients who don't have endometriosis to see what conditions are associated with endometriosis patients. And we do this across both the full patient timeline and only for pre-endometriosis conditions. And then in the second analysis, we cluster our endometriosis patients, dividing them into groups based on the similarity of their comorbidity patterns. And we try to understand what comorbidities drive each of those clusters. Next slide. So here's a more concrete picture of the patients we're looking at. So we're analyzing patients from, first off, I'll note that we do this both with UCSF's electronic health records, as well as UC-wide electronic health records, where we can test which of our associations that we find at UCSF hold up. So at UCSF, we have about 19,000 endometriosis patients, and UC-wide, we have about 24,000. So these are, pretty big numbers, and certainly much bigger than most studies of endometriosis in the past. And then for the control patients, we match so that the distribution of the control patients' age, gender, race, ethnicity, and health utilization is roughly the same as what we see in the endometriosis patients. So that's what is in the gray boxes in the middle, and that's what we compare to for the first analysis. And then the last thing I'll note on this slide is that we have subsets of the endometriosis patients who have records prior to their first diagnosis, and that's who we use for that part of the analysis. So we look at pre-endometriosis conditions. So I'll cut to the, I guess, the main result, which is that we find 661 conditions significantly associated with endometriosis at UCSF across the full patient timeline. So on the vertical axis here, we have conditions organized by ICD chapter. And then on the horizontal axis, we have the strength of the significance of the association. I think the first thing to note is that the associations span almost all of these chapters, almost all of these body systems, right? So this kind of reflects the systemic nature of the condition, as we were talking about before. And if you look at some of the top associations, which I've highlighted with labels, they do, make sense. So we see things like excessive and frequent menstruation, pelvic pain, dysmenorrhea, uterine leiomyoma. These are all the kinds of things we expect to see associated with endometriosis, which is a good sanity check. Next slide. If you dig into the results a little more deeply, we identify several conditions that both align with prior literature, as well as a few others that are less commonly reported. So obviously, there's a lot of discussion about the links between endometriosis and infertility. So we find in our data that endometriosis is pretty strongly associated with infertility. We also see a reflection of the general autoimmune association that I brought up before. In terms of some of the other conditions whose links to endometriosis are maybe less well understood, we see things like migraines, gastroesophageal reflux disease, asthma, and vitamin D deficiency. So these are things that have been talked about like a little bit. You can find a couple of papers on each of these, but not super widely. But despite this, we see pretty strong associations in our endometriosis population. So these could be interesting avenues for further exploration. Next slide. So all of that was at UCSF. And we also did this analysis UC-wide, and we found that 302 of those conditions that we found at UCSF, including the ones I've talked about so far, were also significant in the UC-wide data, which adds some additional support. And if you look at on the right in the plot, if you look at the strength of the associations for the same conditions in the UCSF data versus the UC-wide data, you'll find that they're pretty well correlated, which is reassuring and another good sanity check on this analysis. Next slide. So the next part of this was looking at conditions that are associated with endometriosis before the very first diagnosis. You'll see that this plot looks pretty different from the first one, where we considered conditions across the whole timeline. There's a lot less ICD chapters that have significantly associated conditions, and the significant associations fall mainly into diseases of the genitourinary system. So this does kind of make sense, since these are the sorts of symptoms that endometriosis patients present with before getting their diagnosis. And next slide. And if you look at the overlap with the UC-wide data, we find 34 significant conditions, which is fewer than before, which does make sense since this is all before the first diagnosis. But the correlation between the UCSF and UC-wide data is still good, which is good to see. Next slide. So if you dig into these conditions a little more deeply, we find a number of interesting things. So first off, we see that migraines remain significantly associated with endo before the first diagnosis, which adds kind of another layer to that story. We also see that elevated levels of CA-125 are pretty strongly associated with endometriosis before diagnosis. This is interesting because there's been a fair amount of work done on this marker. The short version is that many people with endometriosis have high levels of CA-125, but not everyone who has high CA-125 has endometriosis. So it's not a great diagnostic tool, but nevertheless, the kind of strength of the association here with an odds ratio of 17.9 is pretty remarkable. And lastly, I wanted to bring up a couple of associations we found on the protective side in the UC-wide data, namely hyperlipidemia and mixed hyperlipidemia. These are interesting because there's been a decent amount of work done recently looking at whether statins, which are the treatments for hyperlipidemia, could be used as therapeutics for endometriosis. So it's pretty interesting that we see these on the protective side with an odds ratio less than 1, meaning that it's associated with the control patients and not the endometriosis patients. So that is another interesting finding that could be something to look into further. Next slide. So I'll be pretty brief about the clustering results. The way to look at this plot is that each node above the dotted line is a cluster in the UCSF data, and each node below the dotted line is a cluster in the UC-wide data. And then the links between the nodes are based on the shared enriched conditions. So for example, the same conditions that were found to be enriched in cluster 20 at UCSF, which were mainly skin disorders, were found to be enriched in cluster 5 in the UC-wide data. So the key takeaway from this plot, at least for me, is that we see two big clusters kind of emerge that are related to cancer and pregnancy. This is not super surprising because both cancer and pregnancy are, very big events in someone's health record. But the fact that it comes out in the sort of unsupervised, unlabeled method is pretty cool. And it does indicate that endometriosis patients with these comorbidities have, pretty unique clinical profiles. Next slide. And to that end, we can do this clustering analysis both before and after the endometriosis diagnosis and see how patients kind of move between clusters. So that's what this plot is showing. On the left, we have all the cluster assignments prior to endometriosis, and on the right, we have the cluster assignments considering the whole patient timeline. And we see that patients who are in clusters defined by hyperlipidemia, mental health conditions, pregnancy, or anemia tend to kind of stick together before and after their endometriosis diagnosis, which indicates that they're kind of experiencing somewhat shared clinical trajectories. So that's, another interesting starting point for further analysis that we could look into. Next slide. So to wrap up and summarize some of this work, we've identified hundreds of conditions that are significantly associated with endometriosis, including many that are associated prior to the first diagnosis. And we find that many of these conditions replicate in a totally distinct data set that comes from 5 other medical centers. So our findings align with prior literature on endometriosis comorbidities, which is a good thing, and then also highlight a number of less characterized conditions. And we find a number of consistent subgroups within the endometriosis patient population. So looking forward, this work lays the foundation for future studies exploring further relationships between endometriosis and its comorbidities, and can help support data-driven and AI approaches to modeling and understanding endometriosis patient trajectories. And yeah, with that, I'll hand it back to Marina to wrap it up.
Thanks so much to both of you. So hopefully you got a flavor of what this data is like and how we can use it in different ways. I want to expand a little bit and just share the magnitude of the clinical data that we have access to at UCSF and beyond. So this is a resource that has been championed by the Baker Institute at UCSF called Information Commons. And Boris, who I believe is on the call, is one of the folks who contributed significantly to it. The whole effort has been driven by Atul Butte over the past 10 years, and it contains data on millions of patients. We also have access to de-identified clinical notes. We have imaging information as well as radiology report and some limited genomics, mostly in the context of cancer. All this data is de-identified. It's AI ready and accessible using many of the tools, computational tools and packages that are mentioned on the bottom of the page. We also, as Umair alluded to, have access to the electronic medical records data across the UC system, which is broader. It's not as deep as the data that we have at UCSF, but an incredible resource. And as you can see by the study design that Umair demonstrated, that we can use UCSF data as discovery and be able to validate our findings, whether they're associations, clusters, or predictive models across the whole UC system. We're still working on the identified clinical notes and radiology images UC-wide, even though we have that data at UCSF. And finally, we can now interact and use some of the recent large language models on UCSF clinical records data. So this is effort by the ARS IT group, specifically Kila and his team. Versa is a tool that folks at UCSF can get access to and interact with many of the data sets that we mentioned through a chat or an API. This is HIPAA compliant and available to all UCSF faculty, staff, and students. So hopefully today we gave you a flavor of how we can use data, whether it's molecular data, which was used in the context of drug repurposing, or clinical data, looking at comorbidities and patient trajectories and clusters, as well as computation and AI to try and better understand endometriosis and get a little bit closer to the promise of precision medicine for women with this devastating disease. With that, I'd like to thank all the members of our team here on the left-hand side. This is my lab, of course, Tomiko and Umair, as well as all of our collaborators. the Judice Lab for collaboration across many, many years. We work very closely with several folks at Stanford, Brice Goodnier, David Stevenson, Nima Agapur, and their respective groups, as well as many other collaborators across the world. Our funding sources, this work is funded by NICHD, and we're happy to take any questions.
Thank you so much for this amazing review and incredible presentation. It's really exciting to see all the ways in which AI is being used. I don't have any questions yet in the chat about this, but I have a couple questions of my own. I was wondering if we could discuss real quick, how is AI being used for specific to the diagnostic and therapeutics in the management of endometriosis and in the evaluation?
That's a great
start.
Yeah, I just wanted to, we didn't mention it specifically during this presentation, but we've started to sort of make a foray into leveraging large language models and having a better understanding of patients' experience with endometriosis. Specifically, we took publicly available social media data, and as Marina mentioned, that tool Versa is available to those of us at UCSF. And we looked at trying to understand the patient's experience with healthcare, specifically, treatments and what individuals have been recommended. And it was interesting to see what was identified from, it was many years, it was thousands and thousands of posts. And it was interesting to see that this analysis, which if we had done it manually, would have probably taken years, but using large language models and a fairly robust, computational platform, I think it was done in about, I don't know, a couple of minutes. I mean, it took some while to wrangle the data. But anyway, just having the large language model analyze this data took not that long. And it recapitulated some of the, what has previously been, I guess, known that patients experience. There were some things that I guess are lesser known and somewhat surprising, but still, it's a way of for us to, I guess, explore a lot of data in a different way than we have been doing. And so you can imagine, in addition to leveraging this kind of more publicly available data, we might try turning it towards maybe some of this data that we have available through UCSF, like the electronic medical records, and seeing how that might help with better understanding the disease. So I don't know if Marina and Umair have anything else.
Umair, do you have any thoughts?
No, I mean, I think Tomiko covered it great there. I mean, I think there's kind of two sides of it. There's like, what do we as researchers use? And then there's also like, what do patients use? So I think Tomiko just outlined, we can use AI to look at much more data, much more quickly, and in much more interesting ways than we ever have before. But I also think it's super exciting for sort of patients to have access to, tools that they didn't have access to before, especially when it comes to diseases like endometriosis that are sort of understudied and under-recognized. So I think there's kind of two sides to that.
Absolutely.
Well, I would say there's probably a third side to it where clinicians are able to use AI. And I'm not a clinician by training, I'm a researcher, but I know one of the things or tools that has been implemented at UCSF recently is ambient listening and note-taking for all clinicians. And I've witnessed it, hands-on, being at a doctor's appointment with my mom and having the clinician ask, well, can I use my phone and ambient listening to take notes instead of me typing? And the experience was drastically different, having an interaction with a clinician 100% without them looking at a computer during the visit was pretty drastically different from what it was before. So I think AI is impacting different parts of our lives, whether it's research and making our research more efficient and effective, or making the clinical interactions more effective, or allowing patients to access information that they haven't been able to access before.
Certainly. Yeah. There's a lot of ways AI is coming up and benefiting everybody. So that's exciting.
Yeah, and I'm
What
sorry,
are,
one more comment on
yeah,
that. I mean, you can also think about it. AI is great, but also non-AI approaches, traditional statistical approaches, like the drug repurposing work that Tomiko presented. I wouldn't say that's AI. That's just, statistics and data science can be very effective in generating hypotheses too. Building off of Omer's work, we can think about now as the next step, building machine learning models to predict which patients are more likely to develop endometriosis given their pre-diagnosis profiles. So you can see how you can use different approaches to ask different questions, and they can be impactful in different ways.
yeah, absolutely. What are the benefits and drawbacks of leveraging public data?
Well, I can take that one. I can start at least. So we leverage a lot of public data in our work from the lab, I would say in terms of my research lab. And there are benefits and drawbacks to it. The benefits are that, and I would say the electronic medical records data is not public. This is data that's internal to the university. We can use it for research purposes, but it's not out there on the internet. Tomiko was talking about an example of using Reddit data that's publicly available. And I think there are tremendous benefits to being able to use this free data to ask questions. And it enables researchers who maybe are computer scientists or statisticians who might not have a biological or clinical background to ask questions and to play with these data sets to build models. And can be quite effective. In another area of research that we lead, specifically pregnancy outcomes and preterm birth, we launched a crowdsourcing challenge where we made a large compendium of microbiome data publicly available, and we had researchers from all over the world build machine learning models to predict women at a high risk for preterm birth. So this type of data democratization can be, I think, very innovative and lead to innovative hypotheses. That's the good thing. That's the benefit. It's free. There's a lot of it. And it can lead to interesting questions and hypotheses. I guess the negative aspect is the fact that it's messy. It's complicated. It's not well annotated. A public data set or a public cohort will never be as well phenotyped as, for instance, a cohort that we put together with Dr. Jadis, who knows these patients and sees them in the clinic and knows every little detail about them. So there are benefits and drawbacks to each approach, and we try to synergize and use public data in conjunction with well-curated data sets and put it all together to better understand disease and help patients.
Yeah, I'd like to piggyback great points that Marina raised. I think a few other things that we might consider with publicly available data is if we're able to aggregate them, then we may have statistical power to find things that maybe the individual data sets, weren't able to find. And I think that was done with the meta-analysis of the microbiome data that was led by Marina and Edith Kosti, a former postdoc, that took several microbiome data sets and came up with identified some organisms that had not previously been associated with preterm birth, which was interesting work. Another thing is that I guess if you're using this data that comes from many different sources, then the findings that you have will be more applicable to a larger, population of people, which is also nice, and cost too. Like it's really expensive to come up with these data sets. So if you're able to recycle, and especially for some of these, people that maybe are just starting out, being able to use publicly available data is great. But as Marina mentioned, data is very messy. So that is a challenge, but still, it's a great resource.
Kind of, I guess, piggybacking on this topic, though, what about, can you comment on bias and the risks associated with bias that's in the AI and how that can influence this as well?
Absolutely.
Definitely.
Does either of you want to start?
I mean, I can start a little bit, I guess. So, I mean, I think over the last decade, we've seen a lot of instances where models that are trained on publicly available data sort of reflect the biases of that publicly available data. So I think it's super important, and as we as researchers continue to develop these models and techniques, that we think really carefully about uh, how we evaluate them and how we use them. Something that is trained on public data is always going to be prone to the biases of that public data. So I think there always needs to be an aspect of critical consideration from the teams developing the models about, how can we train the model in a way that it's equitable and doesn't reflect biases that we see in public data. There are definitely ways to do that. I know from my computer science training, there's lots and lots of papers about, mathematical techniques, statistical techniques to make sure that models are trained fairly. I think some of the challenges like getting that literature into like the clinical and biological fields, which I hope to help with. But the tools are definitely there. I think it's just a matter of like applying them.
Yeah, thanks.
Mhm.
Did you guys want to add anything?
I guess I just wanted to. Oh, go ahead, Marina. Did you want to add
No,
something?
go ahead.
Oh, I was thinking about our dream challenge and how, we had people like we aggregate all these publicly available data sets. But one of the things we were really careful to do is include a validation data set that was very different, that did not reflect the population that we had participants train on. And by the way, I just realized it's a dream challenge without explaining what that is. What we did was we made a data set available and posed some challenges that we wanted individuals who would participate in this challenge of ours to try to address. And literally, we took 9 publicly available data sets of microbiome data, shared this with challenge participants, and asked them to be able to predict preterm birth, individuals who were at risk of preterm birth from this data versus those who delivered term, as well as predicting early preterm birth. And so if we had kind of like used similar representation in the validation data set, which we were using to assess the challenge participants, like that wouldn't be good. So anyway, we ended up very carefully identifying individuals who weren't represented to make sure that there wasn't any kind of bias that way. Yeah, it's difficult, it's challenging, but it is something that has to be taken into consideration when doing this kind of work.
Definitely. I don't have any additional questions. Was there any additional comments or anything that you guys are thinking of last minute before we wrap up?
I just wanted to say thank you for your time and thank you everybody for attending. It was fun to talk about the type of work that we do, and we hope that we enticed you, that you can use data in different ways to ask questions and help patients with endometriosis, which is what we aim to do.
Definitely. Amazing. Well, I'd like to thank all of our speakers that we've had tonight and over the course of the series. I'd like to thank all the attendees and for taking the time. Special thanks to Maru, who is behind the scenes helping out as well. We could not have put this on without you. If anybody is interested, hopefully we'll have the recording accessible on our ENACT website for your reference. We hope to see you all in the future. Have a lovely evening.
Thanks so much. Have a good night.
Take
Thank
care.
you.
All right.